Here we have an assessment standard question on herd immunity. The first question is set to level A, the second question is set to level C. Take your time to answer the question. Explain why this level of immunisation protected the whole population. We're looking at individuals that had not been immunised, were unlikely to come into contact with infected individuals, basically the principle behind herd immunity. One reason why it's difficult to immunise 100% of a population, due to malnutrition, poverty, rejection by some of the population, lack of education, lack of access to the medical resources or vaccines, or geographical remoteness. In this PowerPoint, we are going to be focusing on page 37 of your student guide and looking at what we call antigenic variation. By the end of this PowerPoint, we have one single learning intention, and our aim is to describe what is meant by antigenic variation with reference to the role and impact in influenza. We have been looking at what we mean by immunisation, which is the process by which a person develops immunity to a disease-causing organism. This can either be through an active form, whereby the individual suffers the disease and fights it by themselves, or through the process of vaccination, where they artificially are exposed to the antigen, which initiates an immune response, and from this some B cells and T cells persist in the body as memory cells, providing immunological memory. Vaccination is the method of immunisation by which a weakened or altered form of the pathogen or its toxin is deliberately introduced into the body. This vaccine acts as an antigen and initiates the immune response. The antibodies that are produced and the T cells that respond to the antigen in the vaccine persist in the body as memory cells. And if the person is exposed to the normal causing antigen at a later date, it initiates a secondary response. Within a population of pathogenic microorganisms, new strains arise continuously by mutation. These new strains are described as demonstrating antigenic variation if they have antigens on their surface that are different from the original strain. A new strain of the pathogen showing antigenic variation is genetically and immunologically distinct from its parent strain and succeeds because it enjoys a selective advantage. If a virus appears in our system that has a surface antigen, our body's immune response is to develop antibodies that are made in response to a particular antigen. These memory cells are produced and are retained within the body to fight any future infections. If the identical strain of the same virus enters the body, our memory cells recognise the antigens on the virus and trigger an antibody production immediately and would prevent reinfection. However, if a newly mutated strain of the same virus arrives, the new versions of the antigen on the surface of the virus are not recognised by the memory cells, and therefore these new antigens would induce a new immunological response, and the individual would be suffering from symptoms again. With antigenic variation, because of the change in the antigens on the surface of a virus, we find that previously made antibodies will no longer be complementary to the antigen on the surface of the pathogen. Production of new antigens enables the influenza virus, for example, to avoid the effects of the human body's immunological memory. This allows it to reinfect the person because its new antigens are not recognised by their memory cells. It is for this reason that influenza remains a major public health problem. At-risk individuals need to be vaccinated every year with a new version of the vaccine to give protection. Production of new antigens enable the influenza virus to avoid the effects of the human body's immunological memory. This allows it to reinfect the person because its new antigens are not recognised by their memory cells. Antigenic variation occurs in the influenza virus, explaining why it remains a major public health problem and why individuals who are at risk require to be vaccinated every year. Here we have an assessment standards question on antigenic variation. The virus that causes influenza can evade the specific immune response by what? A, B, C or D? The correct answer is D, by showing antigenic variation. That's us at the end of this particular chapter. You are invited to evaluate your own learning against the learning intention shown on the screen. If there's anything you don't understand about antigenic variation and influenza, please revisit the appropriate parts of the PowerPoint. That's us at the end of this key area of immunisation. On page 38, you have your completing the blanks summary, which is set to level C. On page 39, you have your completed summary in line with the SQA document. And again, this is everything set at level C that you need to know for the exam. We have our retrieval practice statements on page 40. Our retrieval practice mind map on page 41. And in class or on Teams, you have your immunisation targeted past paper questions.